Kastrup. So Dr. Christian Kastrup is a senior investigator at Versiti Blood Research Institute and a professor of medicine, or professor at the Medical College of Wisconsin. He's also an affiliate professor here at UBC. He's actually, he was here for 10 years, something like 10 years, and then we lost him to Wisconsin, but he's maintained a healthy presence here, fortunately. He trained in chemistry, got his PhD at the University of Chicago, did a postdoc at MIT, and then came to the Michael Smith Labs from there, where he truly flourished from his science, from his entrepreneurship. He's established several companies. He somehow manages to do all that and then supervise many, many trainees at all levels. He's really a force. And so we are pleased to have him back today. And he's going to give us our first address today. Christian.
Yeah, thank you, Ed. Yeah, thank you all for allowing me the opportunity to come back and be able to share the work that we've been doing mostly at UBC and in Wisconsin. And the story I'll tell you about, there's many people here that have contributed to it, and I'm excited, particularly today, to tell you about it because we're honoring a lot of the contributions that Dana Devine and Don Brooks made, and Dana played an important role in the research I'll tell you about. And, so Ed mentioned we've started a couple companies. I'm required to make these disclosures. The problem that we're trying to solve is problems related to severe bleeding and trauma. And so we can relate to bleeding in many ways. In the picture here is an example of bleeding and traumatic hemorrhage. And this is a big issue. And so the number one cause of death worldwide for young people, and young people I just mean below the age of 45, is trauma. And bleeding is an important component of that. To give you a sense of the size, the magnitude of this, there's about 2 million platelet transfusions that are given annually in the U.S. So that's platelets are given to prevent bleeds and to stop bleeding that's already occurring. And so there's 2 million of these. So there's a lot of bleeding that's happening. So with all the platelets that are given, with all the deaths that are still occurring from hemorrhage, there's an opportunity to decrease mortality from hemorrhage, and that's if we could somehow enhance platelets. So we could create better platelets that were more effective at stopping bleeds, or platelets that are more effective in other indications because they play such a large role in biology and the vasculature. If we can improve them, then it might be able to have a beneficial effect. The problem with this is that platelets are very challenging to genetically modify. So they don't have nuclei, so you can't use the same types of methods you would use for most cells to genetically modify them to express proteins. And they're really sensitive cells to work with. So what I'll tell you about today, and start to tell you about in a few slides, are ways that we can use mRNA to transfect platelets in order to make proteins. And these are in donor platelets, so bags of platelets. But before I do that, just to give you a sense of the types of problems that we're working towards solving, I won't tell you that we've solved this particular problem today with the platelets we're creating, but this is an example of the type of situation. And so what we're looking at here is the survival of a pig model. These pigs have a non-compressible abdominal hemorrhage, so they're bleeding into their abdomen, and it can't be compressed. And in this particular injury, all the pigs have died in about 30 minutes. And this is similar to what can happen in humans with a severe blunt force or penetrating injury into their abdomen. And so really the goal is to extend survival. And we don't necessarily need to take this survival from 30 minutes to 100 years. What we really need to do is just shift this curve up and to the right and really just buy time. Buy time in order to get the patient to surgery where this injury can be definitively stopped. And so I'll tell you about what we're doing with platelets towards achieving this goal. And so we've been developing methods to genetically modify platelets with mRNA. The hypothesis is that we could take donor platelets, so platelets that were donated to Canadian Blood Services or to NetCat or to the Versiti Blood Center, and transfect them with mRNA, and that we could use lipid nanoparticles to help contain the mRNA and keep it stable as we transfect these cells. So that was the idea. So we take mRNA in lipid nanoparticles, or LNPs, add it to the bag. The LNPs would get taken up by the platelets. They would express proteins that could be therapeutically useful. And so, for those of you that know research well since 2011, you'll know that this was took a very long time to achieve. There are many ups and downs, but we have been able to do this, to express proteins. And so this work was really led by Jerry Leong and Colton Strong, Katie Badior, Emma Kang. So, and I really want to point out that Dana Devine was just a major contributor. Help lead this. Stuck with my lab in this project for a long time and help us get there. So there's a couple different ways you can modify platelets, and I just wanted to tell you a little bit why I think that it's useful to directly transfect donor platelets. One is that it's potentially compatible with blood banking processes. So when platelets are collected at the blood center, they potentially could be modified right at that time to express proteins before they're sent to the hospital. It's also, these platelets are more available than platelets that you could grow in the lab. So the platelet-like particles that you can grow from stem cells, right now it's still very limited, the number that you can do. But in that technology, you can potentially make a wide variety of genetic modifications. And of course, if we make modified platelets in this way, it can be faster to administer than a bone marrow transplant. So you can do a bone marrow transplant with modified stem cells that make karyocytes in order to produce genetically modified platelets in vivo. But of course, you have to do a bone marrow transplant for that. So our approach to creating these platelet cell therapies is they really had to optimize the lipids that in these lipid nanoparticles. We had to optimize the mRNA sequence, and we had to combine these efforts in order to enable functional changes in the platelets. And so I'll tell you a little bit about lipids and lipid nanoparticles. And so lipid nanoparticles, they're very effective at delivering mRNA into cells. And Vancouver and the biotechnology sector and UBC played a huge role in creating the lipid nanoparticles that are used in the COVID vaccines. And so we use lipid formulations that were developed by Peter Cullis at UBC and Marco Cifalini at UBC, which are very similar to those that have been approved. And in these LNPs, there's a variety of lipids that are used, and one of them is particularly important. They're all important, but one in particular are these ionizable lipids. And so these ionizable lipids are, under acidic conditions, are protonated and will bind nucleic acids. And then when you bring them up to a physiological pH before you inject them, they become neutral on their surface, and so they're no longer toxic because you can't administer positively charged lipids. There's no positively charged lipids, cationic lipids in human biology. And here's just an example of one. This is called ALC0315, and there's a tertiary amine that'll do this. It'll be protonated under acidic conditions and neutral at physiological conditions. But I'd like to show this molecule and point it out. This is the molecule that's in the Pfizer vaccine, but it's called ALC, and the C actually stands for Canada. It was made in Vancouver. And so platelets, they really require these good transfection agents. And so what I'm showing here is what happens if you take a naked mRNA and you deliver it to platelets, and you compare that to if there's no mRNA at all or no LNPs. And so there's very little protein expression. Here we're trying to make a reporter protein, nanoluciferase, but there's no expression if you just take a naked mRNA. If you use commercial transfection agents like lipofectamine or ribojuice, it's still the same thing. You don't see any expression of this reporter protein. You can get a lot of mRNA into the platelets, though. So the platelets are now full of mRNA, but there's no translation occurring. And then if we use what I'm calling here is pre-optimized lipid nanoparticles, we did there was a whole PhD that went into creating the pre-optimized, but when we use the pre-optimized formulation, we could start to see protein expression in the platelets. And then as we optimize this further by modifying the lipids, we could see another tenfold increase in protein expression. And it doesn't actually take getting a lot of mRNA into the platelets. It's just using the right lipid nanoparticles that can release the mRNA from the right time during the maturation of the endosomes. And so here's just a little bit about the sort of the screening that we had to do to test different lipids. I won't spend a lot of time on this, but the platelets are really sensitive cells. They become activated really easily. They're really trying to, I think, suppress translation as much as they can. So each of these ionizable lipids normally work really well for cells in vitro and in vivo, but some of them don't work at all with platelets. They either activate them or they just don't allow the mRNA to get released and find the ribosomes. And so we optimized the ionizable lipid and the helper lipids. These are phosphatidylcholines. And we had to make combinations of these to find some that would really, the platelet would transfect the platelets and allow them to make proteins. And there's a couple of things that are really interesting is that the protein expression didn't correlate with platelet activation, and it didn't even correlate with the amount of uptake of mRNA. So platelets don't express proteins very much, but they do have inducible protein expression when they become activated. So there's a few proteins that are known that will start to be expressed in the platelets when they're activated. But in this case, with the mRNA we're delivering, that's not what's happening. So the platelets don't need to be activated. In fact, there's no correlation between the amount of protein that's made and their activation level. And so that was we had to do to optimize these lipid nanoparticles, and we think it's relevant to other blood cells as well, potentially useful for other types of cell therapies. Then we had to optimize the mRNA. And so platelets, they have really interesting mRNA sequences that really seem to repress translation. And so we had to optimize many aspects of the mRNA. Here's just one example of that Madeline Robertson, a PhD student in the lab, did. And so this is looking at the untranslated region of the mRNA and looking at different types of UTRs, 5' UTRs. And depending on how we change that 5' UTR, it has a big effect on whether the platelet will turn that mRNA into protein or not. And so this is comparing megakaryoblast, so like precursor cells to platelets, to platelets, as well as looking at hepatocytes. It's just like another cell type that's. And so what I really want to point out is that platelets really are special in terms of translation. And so in the data that I'm highlighting in the box, there's two different UTRs, and they have but these UTRs are very similar expression levels in hepatocytes. So the hepatocytes don't seem to care too much which UTR that it is. And in megakaryoblast, this UTR3 has a higher expression than in the other UTR. But in platelets, it's reversed, and there's a really big effect. So platelets seem to be special. And so we're trying to figure out all the mechanisms that are occurring in these processes, and I'd say we're at an early stage in that. And so now I'll tell you about being able to take these platelets and testing if they can be transfused and if they can contribute to hemostasis. And so these mRNA transfected platelets, they can still respond really well to agonists. And we selected certain lipid nanoparticles and mRNA that would make it so that they would not be readily activated by the transfection alone. And so without adding any agonist to the platelets, just using platelets themselves, the lipid nanoparticles, after a lot of optimization, they don't induce activation. So we don't see CD62P on the surface of the platelet, so they're not getting activated. But if we add an agonist like ADP or thrombin or others, we get a very robust activation. So the platelets still seem to be doing their job in this case. And if we look at if the platelets can still contribute to blood clots ex vivo, here we're looking at a thromboelastography using ROTEM. So whether or not the platelets are expressing proteins, they can still help to initiate, grow, and stabilize the blood clot. So there's no difference in the clotting parameters ex vivo. So we wanted to see if we could transfuse these into animals. And so there were collaborators at the U.S. Army Institute of Surgical Research, so Andre Kapp and members of his team, and they had a rat model that they used to assess circulation of platelets and to assess the influence on bleeding. And so we so in order to do this in the rat model, we had to test if we could transfect rat platelets, and so we could. It needed different lipids, which was interesting, and Dana told us many times, rat platelets are very different than human platelets, and so she's definitely right in this case. But we could transfect rat platelets. And so we used a rat model of polytrauma because this was a model they already had set up, and it could give us an indicator of whether these platelets would influence bleeding at all. And so, so we transfused untransfected or control platelets. We transfused the transfected platelets. And what we were hoping for, expecting, is that there wouldn't be any difference in bleeding. So if there was excess bleeding, then maybe the platelets were dysfunctional. If there was very little bleeding, if there was fast clotting compared to the controls, that'd be a bad thing. Maybe these platelets had some thrombotic risk. So, so first we looked if they would still, after transfusion, if they could still circulate. And so we labeled them and looked at circulation just for 10 minutes, so not very long. But that would still give us a good marker because platelets can be rapidly cleared. Like if you chill platelets down, if you have a cold stored platelets, they're cleared within minutes. And so we did this, we labeled them, and we transfused them. They were diluted quite a bit by the time they actually made it into the rat. And so in the green dashed line is what we'd expect if there was no clearance, and that's what we saw after 10 minutes. So there probably was a clearance of the platelets, but some clearance, but we couldn't detect it. So we know there's still quite a bit of these edited platelets still circulating at that time. And so we looked at injury. So there was an injury to the kidney that was done in these rats, and we looked to see if there are any changes in the hemostasis time, how fast the bleeding would be stopped. And we didn't see any difference. So whether the platelets were expressing this reporter luciferase, whether they weren't, we got the same bleeding times, which is what we were expecting to see. But what's really neat is that we could see these transfected platelets accumulating at the site of injury. And so in these histology slides, in the blue are the cells in the kidney, and there's a cut that's made. It kind of looks like a triangle, and then we can see the platelets coating that area. And in yellow are these transfected, these edited platelets that have accumulated there. And so what I really want to point out here is that we're showing is that there's local delivery of mRNA and protein from the platelets to that site of injury, which we're really excited about, because it's normally really hard to get a delivery of RNA. In RNA therapeutics, it's hard to get it delivered to areas outside of the liver. And so here we're showing that we can deliver it right to the site of this vascular injury. And so this is a major current direction of where we're taking this, is to be able to take platelets, load them with mRNA, and test if they can be transferred to tissues in vivo, to transfer their mRNA to locally transfect places that are hard to transfect. And what we hope is that we can, this can lead to a platform technology to improve transfusions, target therapeutics to areas of vascular damage, and as a research tool to understand protein expression, understand translation in platelets, and where platelets are naturally delivering their contents to. And so, and I have a little bit more data, but really, in the future, what we're trying to do is be able to control protein production in all tissues. And so in this slide, this isn't a platelet-mediated delivery of RNA, but it's the type of thing that we're working towards. And so in this case, we're delivering mRNA with lipid nanoparticles systemically in pigs, and trying to understand where this mRNA is going and where protein is being translated. And what we can find is that there's actually mRNA being delivered to a wide variety of tissues. And so this is looking at the bone marrow. And so we can detect this reporter protein in hematopoietic stem cells, HSCs. We can detect it in megakaryocytes and platelets in the bone marrow. And we can detect it in the blood as well. Now, if I showed you the amount of protein expression that was occurring in the liver, these numbers would look really, really tiny. So we're not seeing a lot of protein expression, but we're seeing some. And we think that by using platelets, we can increase this going forward. And so just one more data slide after this one. What we're really trying to do too is figure out ways to implement this at blood centers. And so the question is, can platelets be transfected within clinically relevant storage solutions, such as a plasma or platelet additive solutions, so PAS? And so this is something that Colton and Emma and Jerry in the lab have been working really hard on. So the problem that we had, what we've been doing is transfecting the platelets when they were in like a crystalloid, a buffered solution, like a Tyrodes, which isn't compatible with what Canadian Blood Services or the Versiti Blood Center would normally do. And so we were using one lipid that was really good in these buffers, and when we moved it into plasma, it wouldn't transfect the platelets in plasma. So what we had to do is transfect in a buffer, and then like 10 minutes later, move into plasma, which would be complicated. But by screening many different lipids, libraries they have available, such as from Nanovation Therapeutics that I have a conflict of interest with, just to disclose, but we can find lipids that we can transfect platelets even while they're in plasma. And that decreases the amount of the negative effects in the platelets even more by having them in the plasma. And so we can transfect in a PAS to plasma with a plasma ratio of 30%, which is a commonly used solution in transfusion medicine, and in 100% plasma. And so, so in summary, I like to say, what would you like expressed in your platelets? But I do have to say, this works really well with reporter proteins. We're not making a lot of protein in these platelets yet. So if I think about all the proteins in platelets and rank them from like the most abundant proteins being in the 99th percentile and the lowest abundant platelet proteins in the first percentile, we're in about the 4th or 5th percentile. So it's physiologically relevant in some ways, but it's pretty low concentrations. But we're continuing to optimize this. And so, as I mentioned, we hope to use these for transfusion medicine, potentially for targeted therapeutics, and as a research tool. And so, I really want to highlight all the collaborators that are involved, Peter Cullis, and especially Dana Devine and Eric Jan, and the trainees, especially those that are here today, and Madeline online. This is my group in Wisconsin. Some of them are from UBC, and others that are working on this project as well. So, so thank you very much again for the opportunity to come back and share our work. Thanks.
And we have questions open to anybody? Hey, right. Watch Alex and
So this might be a really stupid question, Christian. So how is this technology going to work or not work with pathogen-reduced platelets? Now everything in Canada and United States is Sorlin treated, UV light. Is it going to somehow obviate this technology, or is it compatible with it?
Right. Yeah, that's a great question. It's something that so we're trying to get a PRT, so pathogen-reduced platelets, to test that. And so I think the big question for us there is, would we transfect before or after that PRT therapy? Now that pathogen reduction technique, it degrades nucleic acids. That's sort of the purpose of it, and that's what we're doing here. We're giving the platelets nucleic acids. So I'm assuming that when we do that, when we use that pathogen reduction therapy, that it might degrade all of our RNA and really just kill this at that stage. And so so we'd have to come in after transfect after that. But we still have to test that. But that's a really good question.
While Alex is running, I have a question. Kristen, can you hear me? This isn't working.
Thank you so much, Dr. Kastrup. I have a quick question. Do you think we will see this technology in the clinic soon?
No. I hope so, but it is. There's definitely barriers to overcome. I'd say we're still at an earlier stage for this particular technology. There's other things, but just to compare this to some of the other things in my own lab that we are talking to the FDA on other like hemostatic products that are like gauzes, which are much more straightforward. So here, you're introducing a number of new components into the blood bag that'll be challenging, and being able to even get it to work within sort of the blood system, I think will be a challenge. So if we can have it in clinical trials in 10 years, I think that would be a big win. So I don't know if that's soon or not, but if 10 years is soon, then soon. Yeah.
Nice to see you. I was curious about expressing proteins and platelets that may be normally expressed somewhere like the liver, and the possibility of generating an autoimmunity.
Yeah, right. I think that's a good question. Yeah. So I don't have a great answer to it, except that one thing that is being done, and is there's already I think 2 patients have been tested with this now, is technologies that rely on a bone marrow transplant. And so for patients with hemophilia A, there's an option where you can transfect the bone marrow, make a karyocytes to express factor VIII, do the bone marrow transplant. So then now those patients are now expressing factor VIII in their platelets. And I know in the dog models that were done, the other models, I don't think they saw those reactions, but and the patients haven't experienced that yet. But I do think it's a potential concern.
Christian? So is there any variability, say, from in terms of efficiency of transfection between individuals based on sex, based on age, based on race? And then the second part of that is then, what's the stability? I wasn't really clear on the stability of following transfection. How long does the protein, it may vary with the protein, but how long does expression continue?
Yeah, those are both good questions. So we're trying to figure that out about the donor characteristics and how that influences platelet expression, and other things about the platelets too, like is it young platelets or older platelets from the time that their platelets are formed. And so that one's been tough for us to answer so far, because most of the platelets that we've gotten, they're pooled from 3 donors. And so, but what we've been doing is collaboration, with NetCAD and Ellen and Divine Lab, and is look at a large number of bags of platelets and characterize many aspects of the platelets and try to correlate expression with those things. So it's challenging, but I think we're at 50 bags now, so we should have that data. But it's a tough one that we weren't able to get with 10 or 20 donors so far. And your second, the stability. Yeah, so the protein expression, it'll last a couple days. So we see sort of maximum protein expression in about somewhere around 12 hours. But you can continue to see protein being made and expressed for like 2 or 3 days. We really have to transfect the platelets at the very start, so we can't let the bag sit for a few days. If we let the bag of platelets sit and then transfect, we see a little bit of expression, but not very much. So we know we have to do that. But there are some tricks we can play. We can chill the platelets, so transfect them, chill them, and we can come and warm them up, and we can get a boost of expression days later, up to 7 days at that time. It's not as big of it's not as much protein expression as if we had just done it from the start, but we can sort of do tricks like that. Keep them chilled, and then warm them up to induce the protein expression. Yep.
Any other questions? Going. If not, thanks, Christian.